All right, today we'll be discussing muscles of the physiology course at the Medical University of Wrocław. This part right here in this video will be discussing the structure of the muscle. In general, the muscles can be excited, so action potentials can cause the muscles to change their structure. Contraction, extension, and elasticity are some major characteristics of the muscles. And we can see different types of muscle tissue here. We can see this is the skeletal muscles. These are the cardiac muscles, and these are the smooth muscles. And so we're going to discuss later in those coming up videos on how different muscles contract and the physiological aspect, the very detailed physiological aspect of every contraction process. All right, so in general, this is just a general idea of how the anatomy of a skeletal muscle is. And so it is very summarized into first we got a breakdown of how things are structured here. We have to note down that the skeletal muscle is composed of connective tissue, blood vessels, nerves, and most importantly, the muscle fascicles, which are composed of individual muscle fibers. And then these muscle fibers contain sacrolemma or T-tubules, the sacroplasm, which is the environment that these fibers exist in, and multiple nuclei. And so if we go down, we can see that the sacrolemma T-tubules are part of the sacroplasmic reticulum, which is in charge of storing the calcium that then releases it into these T-tubules that are a major important step in the contraction process. The sacroplasm contains a lot of things. Part of it is sacroplasmic reticulum, the myofibrils, the mitochondria, which is also important for a lot of ATP production, and glycogen granules, because glycogen is needed to be broken down into glucose as a source of energy. Myofibrils are composed of many things here, the troponin molecules, the actin filaments, which are the thin filaments, the tropomyosin, the myosin itself, titin, which is a structural support molecule, and nibulin. Myosin here is the thick filament. And the things that are organized into sacromeres include actually all of those things are part of the sacromere, and I'll show you guys in the future what I'm meaning here. So let's just dive into what actin and myosin molecules are. And so the myosin heads are part of the thick filaments, and so each thick filament consists of myosin molecules whose heads protrude at opposite ends of the filament. So these thick filaments end with myosin heads, and those heads protrude outwards because they have a very important function of being or binding to actin, the actin filaments on those active sites that you guys see here. So a myosin molecule is mainly made up of a tail and a head, and the head has two important sites: the actin binding site, which is here, the tip of it that binds to those circles that we're going to talk on here on the blue balls, and the ATP binding site on the head. And those binding sites are very involved within the contraction cycles. Mainly speaking, the power stroke that we're going to talk about later. The thin filament consists of two strands of actin subunits. And you can see those subunits right here revolved around each other, twisted into a helix, plus two types of regulatory proteins, the troponin and tropomyosin. And we're going to speak about those molecules in details, as they are the ones that regulate the binding of this myosin heads. Because as you see that, just a small information here, that this tropomyosin covers the binding sites of the actins. Okay, and so actin subunits are very different from myosin subunits, and keep that in mind. And so these actin subunits are just joined together to create those strands-like structure. Okay, and so we're gonna then dive into maybe a little bit about how these things are arranged. So you can see that this part right here is the myosin filament, has cross bridges, hinges, and a body. And so the actin filaments are aligned just close so that the myosin heads can attach to those areas that we spoke about just earlier. Okay, and this is just a molecular aspect on how myosin filament would look like. Okay, you can go ahead and read what's written here, but I'm just going to continue. And here, this is a complex that is very important because what this complex does is it gives a more stabilizing function. It's called the dystrophin glycoprotein complex. And so what we're seeing here is that everything that you see starting from the blue rod with this part right here, up here, all of this is the dystrophin glycoprotein complex. And what this does, it binds to actin, right? So it binds to actin. There's this blue rod here, it's called the dystrophin protein. It connects the actin filaments right here to the transmembrane protein B. It's called dystroglycan beta. Okay, and so it connects that in the by a small protein called the syntrophins. So this rod is connected to the beta protein by the syntrophin. Okay, and so it's just explaining to us how things are connected. And so the complex extends its stability by the function of the sacroglycan complexes here. And so this structure outside would connect to extracellular structures. And you can see here, it says this complex adds strength to the muscle by providing a scaffolding for fibers and connecting them to the extracellular environment. And disruption of this can cause many muscular dystrophies. So it just is an important complex for the stabilization of the muscle. And so this is what I was talking about, the sacrotubular system. And so let's go back to the scheme. The sacral lemma or T tubules here. This is what we're here actually. This is exactly what it is. So the sacroplasmic reticulum or the sacrotubular system is made up of a T tubule in the middle with a lot of terminal cisterns of actually not a lot, it's just two. So 1, 2, and the T tubule between them. So those two things on the sides would release the calcium molecules into the T tubule. This combination is called the triad, and this triad is very useful for the activation of sacromeres to cause contraction of the muscle. And so what I'm talking about is this structure here. This is called a sacromere, and this sacromere is what is stimulated by the release of calcium so that it becomes shorter. And so when the sacromeres are shorter, this means that actin and myosin filaments are interacting, and so causing this muscle to shorten. And I'll go into the system in my next video on contraction of muscles, of skeletal muscles. This is what I was talking about. The sorry, there's a lot of cars around. The sacromere. Okay, I think we need to close the window. Just give me a second. All right, so the sacromere is basically a unit where we see a lot of interaction between the myosin heads here and in blue, the actin filaments. And so this sacromere can actually shorten because just imagine right now quickly how if this myosin head has no, these thick filaments that contain a lot of myosin heads binding to the actins in blue. And so imagine those blues get pushed to this direction as the myosin heads get them to closer to each other, like that. So they will bind and pull, bind, pull, bind, pull, and you will see that the sacromere is getting shorter. Okay, and so just to explain to you those zones quickly, if you really need to know, the sacromere is made up of many zones: I, Z, H, A, Z, I. So these zones are very specific for the presence of different filaments. So to start with, zone A is where we see a lot of overlapping between myosin and actin filaments. The H is that we don't see any overlap and we see only thick filaments. I is we see no thick filaments and only thin filaments, and Z is where those thin filaments or actin filaments are connected to each other. Okay, okay, so I will move on, but just to let you guys know that image one is when the sacromere is relaxed. You'll see that the sacromere is in its largest size, and fully contracted sacromere for muscle fiber is quite shortened compared to this one, okay, because of what I just explained. All right, so this is all right. So I'll stop here for this video. Next, we're going to talk about contraction of skeletal muscles. Thank you guys for joining in. I hope this video was very useful for all of you, and let me know if you like this video or if you have any questions. Put them down in the comments below and give me a thumbs up if that's possible. Thank you very much.